Hello students. We'll start with the reaction mechanism today. Okay. So in reaction mechanism, we need to deal with four rearrangements here. One is Beckmann rearrangement. The other one is Favorsky rearrangement. Then comes benzidine rearrangement, and then benzylic acid rearrangements. So we have four rearrangements that we need to discuss here. So the study of conversion of reactants into products is called as reaction mechanism. So reaction mechanism involves different steps from beginning to end. Okay. It also involves intermediates and reagents along with energetics of the reaction. In the study of mechanism, we often come across some of the reaction taking place through some rearrangements of intermediate species. Okay, today we are going to deal with those reactions in which there is some rearrangement taking place. The first rearrangement that we are going to discuss is Beckmann rearrangement. The first rearrangement that we are going to discuss is Beckmann rearrangement. We'll see what is this Beckmann's rearrangement and actually how it takes place. Okay. This rearrangement involves ketoenzymes. Okay. So this is a rearrangement of ketoenzymes into N substituted amides catalyzed by acids. See, what are these ketoenzymes you see here? Ketoenzyme. Keto stands for ketone. So ketone, see that if you have a ketone group, you know what is a ketone group. Okay. Ketones, when treated with hydroxylamine, ketones when treated with hydroxylamine undergoes nucleophilic condensation reaction. That means they undergo a nucleophilic addition followed by elimination. So there will be a nucleophilic condensation reaction. Nucleophilic condensation reaction result in the formation of this compound. R dash. These compounds are called as ketoenzymes. Okay. So Beckmann rearrangement involves the rearrangement of ketoenzymes into N substituted amines in presence of an acid. So what are ketoenzymes? Ketoenzymes are the compounds that are formed by the reactions between ketone and hydroxylamine. When ketone is treated with hydroxylamine, it undergoes nucleophilic condensation reaction. Okay, nucleophilic addition followed by elimination is what it takes place. So resulting in the formation of oximes. So these oximes are called as ketoenzymes. Okay, now let us see what happens in this rearrangement. Whenever a ketoenzyme is treated with H plus, that is in presence of an acid. So migration of group takes place. The group that migrates is found to be the group that is usually trans to this hydroxyl group. Okay, that generally migrates from carbon to nitrogen. Okay, resulting in the formation of this, RCOH. So there is a rearrangement taking place. That is, there is a migration of group from carbon to nitrogen. So actually during the mechanism you will find that this nitrogen will be an electron deficient there. I will tell you why it will be electron deficient there. So this is what generally happens in a Beckmann rearrangement. So it is a rearrangement in which ketoxime is converted into N substituted amides. Okay, later on I will tell you, later on it gets converted into N substituted amides. So this is what first happens. There will be a transfer, that means there will be a migration. Later on this again rearranges. So this oxygen contains lone pair, it moves here, so this moves here, it results in the formation of this compound R C, okay, double bond O, NHR dash. So you get amide, that is N substituted amides is what you get. I will tell you how actually the reaction takes place. So this Beckmann rearrangement involves the migration of group from carbon to nitrogen. Carbon to nitrogen, later on it results in the formation of N substituted amides is what is formed here. Okay, let us deal with the mechanism so that it will be easy for you to understand. So actually what happens in an Beckmann rearrangement is what we are going to discuss now. So mechanism. Mechanism of this reaction. Ketoxime RR dash and OH oxime, this ketoxime in presence of H plus. See what happens now. This H plus protonates this oxygen because this oxygen contains lone pair of electron. Protonation of this oxygen takes place, resulting in the formation of this compound. OH2 plus. Okay, this is formed. Protonation takes place. So there is a formation of a H2, I mean protonation of this oxygen takes place, right? So since protonation takes place, so oxygen donates its electron to hydrogen, so H plus ion, hence oxygen carries a plus charge. Okay, then what happens is, so this oxygen being positively charged, it pulls this electron and it moves out as water molecule. A stable water molecule is lost here. Okay, so minus H2O takes place, water molecule goes off. Okay, resulting in the formation of an electron deficient nitrogen here. So electron deficient nitrogen will be formed here. Okay, see, this is a ketoxime in presence of H plus. First protonation of this takes place, loss of water takes place. Okay, resulting in the formation of electron deficient nitrogen here. Later on, this group migrates to the nitrogen. What happens is here, I'll write it here. So we have R, we have C, we have double bond N. This group migrates. So R, since the R dash has moved here, so this carbon carries a plus charge now. Okay, this group moves, R moves along with the electron deficient nitrogen here. So when this moves, the carbon becomes positively charged. So then what happens? You know that water was initially lost in the reaction. So this water contains oxygen lone pair. With this, it attacks this carbon. So resulting in the formation of an oxygen carbon bond here. So this formed is formed R C double bond N R dash as usual. This water, that is oxygen, hydrogen, hydrogen plus charge. Right, because oxygen has donated electrons, so oxygen will carry a plus charge. Now this oxygen pulls electrons from this hydrogen, resulting in the formation of this compound, that is R C double bond N R dash OH. This is formed here. Okay, this molecule is formed. Okay, this molecule, this contains lone pair of electron. So the electrons moves here, forming a double bond. So carbon valence will be 5. This double bond moves here, forming a negative charge. So anyhow, this has formed a double bond. The H plus, whatever this oxygen, since it moves here, the electron moves here, it forms a double bond. So H plus comes out of this. So this H plus forms bond with this. You get R, C, this double bond O, okay, N, H, R dash. You get an amide. So this is what actually happens in a reaction. So I'll just explain it once again. Ketoxime in presence of an acid, first protonation takes place, loss of water takes place, resulting in the formation of a positively charged nitrogen here. So electron deficient nitrogen, and R group migrates here. So when it migrates here, so the carbon becomes positively charged. So since water is present, that attacks here to form this compound. Then loss of H plus takes place, resulting in the formation of this compound. Then again the electron oxygen moves here to form a double bond. The double bond moves here to create a negative charge. This negative charge takes up this proton to form NHR dash. So an amide is formed here. Okay, so this is the Beckmann rearrangement that takes place. Okay, this is one of the very important rearrangement that occurs. Okay, so there is another important rearrangement, that is Favorsky or Favorsky rearrangement. Let us see what happens in this rearrangement. This is also very simple rearrangement. This is a rearrangement of alpha haloketone into ester in presence of alkoxide ion. So alpha haloketones, they rearrange into esters, okay, in presence of an alkoxide ion. So we'll just take an example of an alpha haloketone. Let us see what is this alpha haloketones first. Now C6H5, CH2, C double bond O, CH2Cl. This is a ketone group. The next carbon is called as alpha carbon. On alpha carbon there is an halogen attached. So this is called as an alpha haloketone. This is an alpha haloketone in presence of an alkoxide ion. In presence of an alkoxide ion, the rearrangement takes place. C6H5, CH2, and ester skeleton is formed. We'll go through the reaction so that you will have a clear idea regarding this. Okay, so C double bond O, O minus, or R is what you can write it as. So you get an ester there. Okay, the rearrangement of alpha haloketone into an ester. The rearrangement of alpha haloketone into an ester is referred as the Favorsky rearrangement. Okay, you can even write it as R also here. Okay, OR. Okay, let us go through the mechanism of this reaction. Okay, mechanism. Okay, you have this molecule C6H5 CH2 C double bond O CH2Cl. This is 1-chloro-3-phenyl propanone. Right. So what happens is, in presence of an alkoxide ion, you know that alkoxide ions are strong bases. So this alkoxide ion abstracts this proton. It removes this proton, resulting in the formation of this compound C6H5 CH minus charge. I've just write it as CH minus charge. You have C double bond O CH2Cl. Okay, alkoxide ion, being a strong base, abstracts this acidic proton. It abstracts this proton, minus ROH takes place. That means OR minus abstracts this proton, minus ROH, it abstracts this proton, resulting in the formation of an carbanion. Okay, a carb anion is formed here. Right? Next, what happens is here, this carbanion, what it does, it attacks this, because this carbon is positively charged because of this chlorine. Chlorine moves out of this. Okay, resulting in the formation, I'll write there, resulting in the formation of this compound C6H5 CH C double bond O. Okay, you have CH2 here. This has attacked here, so the rate of bond formed, Cl goes of this. So this intermediate is formed here. Okay, next what happens? Next, this molecule is attacked by an alkoxide ion. So since you are carrying out the reaction in presence of an alkoxide ion, so this molecule is attacked by an alkoxide ion. So alkoxide ion attacks this carbon, since that is a positively charged carbon. So double bond O moves it, resulting in the formation of this C6H5 CH CH2 double bond O will become O minus, and OR is formed. Right? Now, so next what happens is, this negative charge cannot be longer for on this oxygen, so it moves here to form a double bond. When it moves here to form a double bond, look at the valency of carbon, 1 2 3 4 5. So one bond should break. So this is a 3-membered ring here. 3-membered rings, you know that they are unstable because of the strain. So the bond breaks, resulting in the formation of carbon. So the bond can break here also, the bond can break here also, but if the bond breaks here, whatever the anion formed will be more stable. Okay, this is what is formed here. C6H5 CH minus, bond breaks. You have CH2 here. This is a CH2. This is linked to this C double bond O OR. this anion is resonance stabilized. So if the bond breaks here, whatever the anion formed is not so stable. So the bond breaks in this direction only. Then this takes up a proton from this alcohol and results in the formation of the product, that is C6H5 CH2 CH2 C double bond O OR. So you get an ester. So this is the mechanism that is involved in Feversky reaction. That means an alpha haloketone in presence of alkoxide is converted into ester. This rearrangement is called as Feversky rearrangement. First, what happens? Since this being a base, abstracts a proton, results in the formation of an anion here. This anion goes in an intramolecular attack here, attacks this carbon, expelling Cl, resulting in the formation of a three-membered ring. This alkoxide ion, you have carrying the reaction in alkoxide ion attacks this carbon. When it attacks, the double bond O shifts to single bond O minus. When minus negative charge comes here, one bond should break, this bond breaks, resulting in the formation of an anion. So in presence of an alcohol, it takes up the proton to give you an ester. So there is a rearrangement taking place which results in the formation of an ester. Right? So this is about the two simple reaction. One was the Feversky, one is the Feversky reaction, another one is the Beckmann rearrangement. Thank you.